Welcome everyone. I am Laxmi, BSc 6th semester. Today I am going to discuss about the structure of the chromosome, which is part of paper 6.1 Botany, and we are going to discuss about it, which comes under the cell biology of it. Before directly going to the structure of the chromosome, let's know what is the chromosome. So we have to know that what are chromosomes. Chromosomes are thread-like structure present in the nucleus which carries genetic information from one generation to another. They play a vital role in cell division, heredity, variation, mutation, repair and regeneration. In eukaryotic cell, genetic material is present in the nucleus in chromosomes, which is made up of highly organized DNA molecules with histone proteins supporting the structure. Chromosomes means colored body, that refers to the staining ability by certain dyes. So Cargill Nagli in 1842 first observed the rod-like structure present in the nucleus of the plant cell. And next, W. Waldeyer in 1888 coined the term chromosome. Walter Sutton and Theodore Bowry in 1902 suggested that chromosomes are physical carriers of genes in the eukaryotic cells. The number of chromosomes in any species is constant for all the cells. The number of chromosomes in gametes, that is the egg, that example is the sperms and eggs, is half of the somatic cell and known as haploid set of chromosomes, which is result of the meiosis during sexual reproduction. Chromosome number is present in the mitotic division of somatic cells, which is required for an organism to grow, repair and regenerate. Chromosome number varies in different species. A nematode species contains only 2 chromosomes in a cell, whereas a protozoan species contains as much as 1600 chromosomes in the cell. Most of the plants and animal species contain 8 to 50 number of chromosomes in its somatic cells. The number of chromosomes does not reflect the complexity of a species. A human cell contains total 23 pair of chromosomes, that is 2n in total. 23 into 2, 46 chromosomes, of which 23 are autosomes and 1 is X chromosome. Karyotyping is a technique to study the structure of chromosome present in a species. Chromosomes are isolated, strained and photographed. This technique is useful in finding out any chromosomal abnormalities. That is, this is a karyotype of a human cell of a male. These all are kind of the chromosomes present in a cell. The displaying of all chromosomes is said to be the karyotype. Now coming to the structure of the chromosomes. Each cell has a pair of each kind of chromosomes known as the homologous chromosomes. Chromosomes are made up of chromatin, which contains a single molecule of DNA and associated proteins. Each chromosome contains hundreds and thousands of genes that can precisely code for several proteins in the cell. Structure of a chromosomes can be best seen during the cell division. So during the cell division we can see the best structure of the chromosome, which all are found in a cell. The main parts of the chromosomes are the chromatids. The first of all is the chromatid. Each chromosome has 2 symmetrical structures called chromatids or sister chromatids, which is visible in mitotic metaphase, and each chromatids contain a single DNA molecule. At the anaphase of mitotic cell division, sister chromatids separate and migrate to the opposite poles. Next is the centromere and the kinetochore. Sister chromatids are joined by the centromere. The spindle fibers during cell divisions are attached at the centromere. The number and the position of the centromere differs in different chromosomes. The centromere is called as the primary constriction, also as primary constriction. Centromere divides the chromosome into two parts. The shorter arm is known as P arm and the longer arm is known as Q arm. The centromere contains a disc-shaped kinetochore which has specific DNA sequences with the special proteins bound to them. The kinetochore provides a center for polymerization of tubulin proteins and assembly of the microtubules. Next is the secondary constriction and the nuclear organizers. Other than chromosomes possesses secondary constrictions. Secondary constrictions can be identified from centromere at anaphase because there is a bending only at the centromere, that is at primary constriction. Secondary constriction, which contains gene to form nuclei, are known as nuclear organizers. The next part is the telomere. The terminal part of a chromosome is known as telomere. Telomere are polar, which prevents the fusion of chromosomal segments. And the next part is the satellite. It is an elongated segment that is sometimes present in the chromosomes at the secondary constrictions. The chromosomes with satellite are known as SAT chromosomes. And next is a chromatin. Chromosome is made up of chromatin. Chromatin is made up of DNA, RNA and proteins. At interphase, chromosomes are visible as thin chromatin fiber present in the nucleoplasm. During cell division, the chromatin fibers condense and chromosomes are visible with distinct features. The darkly stained condensed region of chromatin is known as heterochromatin. It contains tightly packed DNA which is generally inactive. The light stained diffuse region of chromatin is known as the euchromatin. It contains genetically active and loosely packed DNA. At prophase, the chromosomal material is visible as thin filaments known as chromonemata. And at interphase, bead-like structure are visible which are an accumulation of chromatin material called as chromomere. Chromatin with chromomere looks like a necklace with the beads. So these all were the parts of a chromatin. These parts organize themselves in organized form to form a chromosomes, which is the basic hereditary material which passes from one generation to the another generation. This is all about the structure of the chromosome. So thank you and all for watching. Thank you.